GMF-1A3 is an antibody that we've developed against Amphiregulin, which is a novel cell surface target in solid tumor biology. Amphiregulin is a growth factor. It's a ligand for the epidermal growth factor receptor. We first became interested in it in terms of its role in breast development. So it is very important during pubertal breast development, where it's a key proliferative effector of the response to estrogen. The thing with Amphiregulin is it comes back to haunt us much later in life. So it seems to be a key driver, particularly in estrogen receptor positive breast cancer. So in terms of targeting it, we showed a long time ago that there's a rate limiting step, which is the proteolysis of Amphiregulin, which cleaves it at the cell surface, allowing the signaling domain to interact with the EGF receptor. We had the idea that we could go after the transmembrane stalk, so the waste product that's left behind after Amphiregulin is released, which gets internalized into the cell. So we've developed a series of antibodies that selectively recognize the Amphiregulin stalk, so that in cells that are shedding a lot of Amphiregulin, we can recognize them with these antibodies. And we had the idea that conjugating these antibodies with the cytotoxin could be a neat way to develop antibody drug conjugates to could target cells in the body that cleave high levels of Amphiregulin, like breast cancer cells. And our additional studies have shown across a range of solid tumors that in addition to breast cancer, it's particularly highly expressed in colorectal, lung, and prostate cancer. All above 50% have high levels of Amphiregulin cleavage. So while breast cancer has been our initial focus for development, the potential applicability is more broad. So we've completed the preclinical validation in vitro models and shown efficacy against xenografts in immunocompromised mice. So we're now looking for partners to help us move forward with the next steps of Amphiregulin GMF-1A3 development.